Yeah, I'm not going to focus too much on the computational methods that my lab develops. That's probably half of the research in my lab. We continue to develop new algorithms and software, primarily to do genome biology and study the title here. We are really focused on germline mutations and increasingly somatic mutations and their role in driving various traits and the consequences of those mutations. And so I'm gonna focus on a recent paper which studied germline mutation in large pedigrees from Utah. This is a picture of southern Utah. I was just there this weekend. Before I get into the details, I just want to make sure I acknowledge the really incredible grad student, Tom Sassani, who's at the top here, who led this work. This was a collaboration with the chair of my department, Lynn Jordy, down on the bottom, as well as some really key insights from Molly Prosorski at Columbia and Ziwei Gao, who was a postdoc at the time in Jonathan Richard's lab. And then a lot of the methodology was developed by Brent Pedersen. And at the end of the talk, I hope to cover some more recent findings in collaboration with a friend of mine named Richard Coffin in my department. Okay, so in terms of germ cells, every haploid gamete that's developed, created by a man or a woman, is the result of a fresh recombined copy of that individual's diploid genome that they inherited from their parents, right? And I think when you step back and think about it, this is a pretty incredible process, especially, well, I'm focusing today primarily on the male germline. Billions of sperm are created over the lifetime of a man, and each time, we're replicating and recombining to result in a 3.1 billion base pair haploid genome. And what's really incredible is that the fidelity of that replication recombination is extremely high. The mutation rate in the germline is actually quite low. But indeed, mistakes do happen. This is often the result of mismatches during replication or polymerase slippage during replication. And not surprisingly, there's a whole host of enzymes that have evolved over millions of years to maintain the fidelity of the germline by repairing mutations in real time as they occur. So when a germline mutation arises prior to zygote formation, so imagine there's a mutation or a collection of mutations in the sperm cell. That's the sperm that goes on to fertilize this egg. And that now diploid genome, haploid from sperm, haploid from egg, go on to develop one of us. Well, all those mutations are inherently heterozygous in every cell in our body, including soma and all of the germline progenitor cells in our body. And so one of the fundamental questions in human genetics and in population genetics has been trying to understand at what rate these mutations arise and accumulate in humans. And this has been studied in other primates and other organisms as well. The focus of my talk is going to be on human germline mutation. And this interest, frankly, has roots all the way back to Haldane, before we even knew what the molecule of heredity actually was. And Haldane, through basically clever intuition, made an early estimate of what the human mutation rate was. And he postulated that the observed allele frequencies, alleles weren't formally described at that time, but in terms of the frequencies of, say, a disease trait, the frequency at which we observe a disease trait is the result of a balance between mutations, the things that's causing the disease, and purifying selection against them. And so the case study that he focused on was X-linked hemophilia in male Londoners. And I'm not going to go into the details. There's a really beautiful paper on this that I encourage you to read if you haven't. But essentially, through his cleverness, Haldane postulated that the mutation rate per locus per generation was about 2 times 10 to the negative fifth per base pair per generation. Now, if you fast forward today and we actually scale this to the known size of this locus, it turns out that his estimate of human germline mutation rate was only off by about a factor of 2, which is, I think, quite staggering. So why do I care about germline mutation? Well, there's many different reasons. Certainly, germline mutation is fundamental to adaptive evolution the way that we understand it today. So to quote James Crow, without mutation, evolution, at least as we understand it, is impossible. So it's really important in terms of understanding the mechanisms of adaptation and speciation. More central to my research at the Utah Center for Genetic Discovery, one of the things that we focus on a lot is trying to identify the genetic basis of rare diseases. We have a neonatal DNA sequencing program where we sequence babies in the NICU along with their parents, as well as focusing on families with a rare disease. And what we find and what others find, this is a Deciphering Developmental Disorders, or DDD study, primarily led by Matt Hurls in the UK. They found that 42% of their developmental disorder cohort carried pathogenic DNMs, or de novo mutations, in coding sequence. So essentially, they attribute almost half of the diseases that they studied to a germline mutation in coding sequence. And this is sort of a very high resolution study of something that's been known for a long time, namely that germline mutation leads to developmental disorders. But one of the challenges in clinical genetics currently is we only can diagnose about half of rare disease patients, and we also struggle to distinguish de novo mutations that may lead to a disease phenotype versus those that don't. In some cases, it's trivial, it's clear as day, but in many other cases, it's a big focus in my lab, it's not always so clear. So how do we go about measuring germline mutation rates? Well, one of the first attempts to do this, well after Haldane anyway, in the era of DNA sequences, was a really nice study from Nachman and Crowell back in 2000. This is one of the first papers that I was asked to read as a graduate student, actually. And what they did was they took the then relatively new whole draft genome sequences of chimpanzee and human, and they looked at pseudogenes, 18 different pseudogenes that they assumed to be neutral, not under any adaptive or purifying selection. And they compared the DNA sequences of those 18 pseudogenes in chimp and human, and they basically just counted up the differences and divided it by the number of base pairs that were studied. And they estimated a per nucleotide per generation mutation rate of about 2.5 times 10 to the negative 8th. So concretely, talking about like 100 mutations-ish total in an individual. 10 years later, Lynn Jordy in my lab collaborated with Lee Hood to study through direct genome sequencing of families, both parents and a child. And they use that, and basically, so having DNA sequences from the parents and children, they can compare the DNA sequence in the child to the parent, and they're basically asking, where are there alleles present in the child that are not found in either parent? Now that could be one of two things, maybe one of three things. True germline mutations that arose in either sperm or egg, and that's why it wouldn't be seen in DNA collected from mom or dad's blood. It could also be sequencing error, and it could also be mutations that arose spontaneously in the blood lineage of the child. Now they took great lengths to distinguish true germline mutations from sequencing error, but certainly they were unable to unambiguously determine that some of these mutations were not actually arising in the blood of the baby because they only had one tissue to study. But in so doing, they estimated a lower de novo mutation rate, more on the order of about 1.2 to 1.3 times 10 to the negative 8th per nucleotide per generation. So about half the estimate measured by phylogenetic methods from Nachman and Crowell. It turns out that today this is really the consensus, that in human, we expect an average of 70 germline mutations per individual. The majority of those germline mutations come from the male germline, and the minority come from the female germline. I'll talk about that in more detail. So it's, depending upon what study you read, I mean, the consensus is between 70 and 75, and this really gets down into nitty-gritty bioinformatic details about what types of mutations you're counting and what fraction of the genome you actually study. But 72 point mutations per child, about 8 insertion deletion mutations per child, many more short tandem repeat mutations. This is primarily owing to errors arising during replication, so polymerase slips on these homopolymer runs. And then something that you'll probably learn a little bit more from Danny Miller's talk, which is, I think, the next one after me. I was saying earlier, he was here a couple of weeks ago and gave a fantastic seminar, but he really focuses on structural de novo mutations. And we find that there's much fewer of these. So it's about 1 per 6 to 12 live births. So most children do not have a de novo structural mutation. And I think that makes some sense given the thought that, a large scale deletion or duplication is much more likely to lead to a dosage effect in protein coding genes and have a phenotypic consequence that might be lethal. Right, so the estimate is about 70 de novo mutations per children, but this is a figure taken from Johnson et al. In 2017, really nice study from looking at de novo mutations in families from Iceland. And what they show on the x-axis is the number of de novo mutations observed per proband or per child, I guess, that they studied. And what you can see is that, yeah, the mode is about 70, but this is a distribution. There are some individuals with many more, and there are individuals with many fewer de novo mutations. And so this sort of gets broadly at one of the questions I've been fascinated with for a long time, which is what are the random and non-random processes that lead to this distribution in terms of the number of de novo mutations per genome. And more broadly, the questions that we're pursuing in my lab are considering germline mutation rates and patterns. We are really interested in how these rates vary as a function of the sex of the parent, the age of the parent, what types of mechanisms lead to mutation. Are some mechanisms differentially enriched in different contexts? And what are the types of changes that we observe? Are there family of origin effects? And are there developmental timing effects? So this is more talking about mutations that arise post-zygote formation. So I think the questions are fairly simple, and from a high level, characterizing or identifying germline mutations and calculating germline mutation rates is conceptually a fairly straightforward process when you have families. So the standard study design is to sequence DNA, often blood, typically blood, sometimes saliva, but the best we can do today is to get high quality DNA from blood from mom and dad, as well as the child. We do whole genome sequencing, and we essentially just look for differences in the child that are not in the parents, with some important caveats. So one of the things that burns us the most when attempting to predict de novo mutations or germline mutations is the following scenario. So let's imagine that the truth is that this T allele was in fact transmitted to the child from the mom. So that's the truth. But when we go to sequence the DNA, we don't know what the truth is a priori. We just take the sequencing data and we're trying to figure out what was inherited and what was not inherited. So imagine if this, in the data that we get back from the whole genome sequencing experiment for dad, mom, and kid, we see the following. Well, we see a mixture of A and T alleles in the child, which would make sense if a de novo T mutation arose, or if the child inherited a T from mom or a T from dad. In this case, the father has lots of A observations, so we're only seeing the A allele, suggesting that the father is homozygous for the A allele at this site. And that, in this case, reflects the truth. Here's where we get burned. Mom, we only see an A allele. We don't see a mixture of A and T allele. And this is because even though we shoot for 30-fold sequencing coverage on average when we do whole genome sequencing or higher, that's a distribution. Some sites in the genome are going to have lower coverage in some individuals just because of random chance. And in this case, we had insufficient depth or insufficient number of observations in mom to recognize the fact that she actually harbors this T allele and it was transmitted to the kid. So in this case, this pattern would fool us into believing that the T allele was a potential germline mutation that arose on either sperm or egg. If we had had greater sequencing coverage in mom, we might have realized that in fact was a false prediction. All that to say that one of the key strategies to developing high quality germline mutation predictions is to sequence deeply as you can afford in mom and dad, so that we can avoid this situation of having insufficient observations or insufficient data to recognize that a variant was transmitted. And this is critical because, we expect these mutations are rare. So going into this, we're expecting that the vast majority of things that we predict to be germline mutations are in fact not. We're missing this scenario. And then the other reason that germline mutation detection is hard is that the human genome sequence that we use, the reference genome, isn't perfect. There are difficulties with short DNA sequences in terms of alignment, and these DNA sequencing technologies that we use, while they're amazing, they still make errors. So we still have to take strides to separate signal, true de novo mutation from noise. And so one of the strengths in Utah is one of the reasons I love living here is 6 amazing national parks, incredible skiing. But with respect to the focus of today's talk, one of the key strengths for studying germline mutations is large multi-generational pedigrees. So you might have heard of the so-called CEPH pedigrees. That's it's CEPH. That's an acronym in French for the Center for the Study of Human Polymorphism. And in the 80s and 90s, multi-generational pedigrees were collected in Utah, identified in blood collected from pedigrees in Utah and in France. And so the focus of the rest of the talk today is going to be studying germline mutations in large multi-generational pedigrees like this. This was featured in National Geographic, and strangely, I visited my mother's home a few years ago when we started this study and found this issue of National Geographic still on the shelves in our basement. So what do these pedigrees look like? The general structure is like this, where we have all 4 grandparents. We have the one offspring from each side, grandparental side, and then we have multiple offspring in the 3rd generation. So in total, we studied 33 large pedigrees like this, comprised of a total of 603 individuals. And there's a median of 8 grandchildren per family. What's really nice about this study is that we were able to go back to the original blood draws that were taken from all these individuals. Prior studies of germline mutation in the 1,000 Genomes Project and of Ceph pedigrees have used immortalized B cell lines, and that's problematic because it becomes difficult to distinguish true mutations that arose in germline versus mutations that arose as a consequence of serial passaging of these B cell lines over time. So being able to do whole genome sequencing from blood of all these 603 individuals really allowed us greater confidence when trying to identify germline mutations. Right, so why are these multi-generational pedigrees so powerful? Well, let me just walk through an example. Let's imagine we've sequenced the DNA of grandpa and grandma in the first generation, and we've sequenced their genomes very deeply so that we can avoid that lack of, avoid the scenario where we don't recognize the transmission of this T allele from one of the two parents. So we've sequenced deeply in the first generation, giving us more confidence to believe that this T mutation observed in the second generation is in fact a mutation that arose spontaneously and was not transmitted. However, even with the deep sequencing in the first generation of father and mother, we can still be fooled by PCR errors or that are, and specific to this individual, or sequencing errors or problematic things like that. This is where the third generation of the pedigrees come in. If this is a true germline mutation that arose in either the sperm or the egg from the parents, this individual is heterozygous in every cell in their body, including their gametes. So this dad, the second generation dad, should have sperm that are 50% of sperm should have A, 50% of sperm should have T, and therefore, if we look at his offspring, we expect half of the offspring to have this T allele. And so let's take a look at an example. So in the third generation, if there were 8 offspring from this dad and the mother, we would expect roughly 50% of the offspring to have that allele. So obviously it can't be exactly 50% when there's an odd number of offspring, right? So this is very much like a coin flipping problem, binomial. So it's a binomial expectation where the probability of success is reflects the probability of that T allele being transmitted to an offspring is 0.5. So given 8 children in the third generation, assuming that this T allele is a germline mutation, the probability of it not being inherited by any child in the third generation is 0.5 to the 8 coin flips. Probability, same as coming up tails 8 times in a row. And therefore, by complement rule, the probability that if it's real, the probability that's inherited by at least one individual in the third generation is 1 minus 0.5 to the 8th. Broad point here being that this third generation, because of the large number of offspring in the third generation, it gives us unique power to really identify high confidence de novo mutations that arose between the first and second generation. So this is the basic study design. We use this strategy to identify high quality germline mutations in the second generation. And this was work obviously led by, and this was the primary focus of this paper that was mentioned at the beginning. This paper is available on eLife and led by Tom Sassani. So now I'm going to walk you through our primary findings. So using some methods that I don't have time to go into today, we could leverage these pedigrees to also attribute each of these mutations that arose in the second generation to the gamete of origin. We can use these pedigrees to decide, did this T allele come from sperm or from egg? And that allowed us to not only break down on the left-hand box plot here, the distribution of total number of germline mutations observed in all those second generation individuals. We could then break down this total into the number of de novo mutations that came from sperm versus egg. And I think what you can see quite obviously is that about three quarters to 80% of the mutations observed in those second generation individuals came from the grandpaternal gametes, so from sperm. And you can also see that the variance is much higher, and this is primarily driven by the variability in the age of the dads at the time that their child was born, or the egg was fertilized. So, and that's because we know that over time, men continue to produce sperm, and the progenitor cells begin to accumulate mutations themselves. And so the total number of mutations that are observed in the resulting gametes increases as a function of paternal age. Right. So then taking all the mutations that we found attributed to either dad's sperm or mom's egg, we could start to break down these mutations in terms of the mutation type. So how often do we see a C allele mutate to a G allele, or a T allele to a G allele, and so on and so forth. And we can compare this mutation spectra in the male germline versus the female germline. And by and large, what we see is that the mutation spectra is pretty much the same, with two exceptions. What we found was that there's an enrichment of T to G mutations in the male germline, and an enrichment of C to T mutations in the female germline. And I want to emphasize that these C to T mutations are not in a CPG context. So we know that when a cytosine is followed by a guanine in the genome, that this cytosine is often methylated and can spontaneously deaminate. These C to T mutations that are enriched in mom's germline are actually situations where it's not a C followed by a G, it's everything but. The C to G mutation, or C to T mutations in the CPG context, are basically identical. Now, what's an open question, this finding was also observed by that DECODE study that I mentioned and a couple other studies since ours, and it's not clear what drives this differential enrichment of different mutation types in the two different germlines. But it's statistically significant and robust. It's been observed multiple times now. Right. So in total, taking the mutations that we observed in the second generation, the rate that we infer is 1.1 times 10 to the negative 8th per base pair per generation. So there's nothing shocking here, nothing new. This is basically the rate that has been calculated multiple times before. So I'm going to try and now focus on some of the more interesting and new findings from this study design. Not surprisingly, and this also has been known, children of older parents have more de novo mutations. So as I mentioned before, if you look at the parental age of birth, or dad's age of birth, starting at sort of 18 years old all the way up to nearly 50 years old, you can see that the number of de novo mutations passed on to their offspring increases at a rate of about 1.5 additional mutations per dad's age, per year of dad's age rather. And on the maternal side, there's also an increase of about 0.4 additional de novo mutations per year of mom's age. Now this result was a hint at this was observed almost a decade ago, but it was thought that it might be an artifact, and the DECODE study and our study, I think, pretty clearly demonstrate that there is a maternal increase. And the reason it was thought to be an artifact is it's somewhat surprising, because oocytes are created in the mother as she's developing in her own mother's womb. And so, you know, there's not this continuous turnover of the maternal germline. Basically, those cells are already created. In contrast to the sperm lineage, where there's constant cell division. So the current thinking, and Molly Przeworski is really the expert on this. She's published a couple papers in the last year on what's driving this effect. And the thought is that because oocytes are present for so long in the mother, as a function of time, they're exposed to exogenous mutagens, and also all the RNA, all the protein available in that oocyte to repair mutations subsequent to fertilization, they age as well. And so her hypothesis is that really this is a manifestation of older oocytes being a more toxic, I guess, for lack of a better word, environment for fertilization, where mutations that arise immediately after fertilization or present from the sperm coming in are less ably repaired in older oocytes. Right. So this increase in de novo mutations as a function of mom and dad's age is also not new. We've known about this from the DECODE study and prior work from Matt Hurls, and earlier work from small family studies. But what I want to emphasize is that this age effect is really this 1.5 additional mutations per year and 0.4 additional mutations per year is an average of the effects observed from all the 33 different families that we studied. And so one of the open questions has been, to what degree does this rate of mutation increase vary as a function of family of origin? And what's really nice about these multi-generational pedigrees is that we're able to get at this question directly. And that's where this many offspring of the third generation come into play. So what we did for every one of our pedigrees was we counted the number of de novo mutations observed in the third generation. So comparing this child's DNA to the two parents in the second generation, counted those number of mutations, and then ranked or ordered the offspring by birth order. So let's say, as an example, this is real data. I chose it because it's one of the more extreme cases. So this firstborn daughter had 43 germline mutations, whereas this lastborn daughter had 92 germline mutations. And 8 offspring, there's roughly at minimum an 8-year gap between firstborn and lastborn. So this allows us to regress or correlate age with the number of germline mutations for each family separately. That was the result of a nice collaboration with Molly Przeworski's lab, and all the methods were developed by Tom Sassani. And so here's an example. So on the left-hand side is all the families together. And what we're just correlating now is paternal age at birth from all these families as a function of the number of mutations observed in those third generation children. So this is what the average trend from all those families thrown together looks like. On the right-hand side are 4 of the 33 families where we've actually regressed paternal age at birth as a function of germline mutations in just the offspring from these 4 different families separately. And what you can see is that, for instance, this family has a very modest increase in the number of germline mutations as a function of age, whereas this family has a much, a rather extreme, these 2 families have a rather extreme increase in germline mutation as a function of age. And so we applied these methods to all 33 families, and this is what the data look like. And so basically the y-axis here is the slope observed for each family. So there's 33 points on this plot arranged from left to right, so each of the 33 families. And on the y-axis is that slope along with the confidence interval for our estimate of that slope. And you can see that the range here is quite broad. So this family was the first one I showed you, where there's barely a quarter additional mutations per year. And this family is 3.3 additional mutations per year. And sort of this midpoint is the average across all the families. So using this study design, these large pedigrees, we feel confident in saying that there's substantial and significant variability in the effect of parental age on the number of germline mutations observed in children. I think the more interesting follow-up questions are, what are the sources of this variability? You know, could there be genetic differences in DNA damage repair genes that are private to each of these families? I can tell you that we looked closely at this, and we saw no overt loss of function variants in any known DNA damage repair genes in any of the parents. We did see variants of unknown significance in DNA damage repair genes, but we don't know enough about the impact of those genetic changes to attribute them to this variability in DNA, the rate of germline mutation increase. Could this be described by just randomness alone? You know, the slopes that I showed you, they are actually rather symmetric about the mean, and they look roughly like a normal distribution. So I think there's some reason to believe that part of what we're seeing is just normal, you know, randomness in mutations. One thing that sort of goes against that hypothesis, though, is if it were random, you'd expect it to be all over the place within a given family, but you can see that there's this continuous increase, like the trend continues for each family. So that suggests that it's more than randomness alone. It could be differential environmental exposures. So one of the things that we are excited to look at is that some of the grandparents in the first generation, you know, a lot of them, these families are all from Utah, and a lot of the grandparents were around in the 30s, 40s, and 50s. 40s and 50s particularly interesting because of all the atomic weapon testing in Western Nevada. And it was known that there's a lot of sightseeing. People would drive out to the border between Utah and Nevada and literally watch from a distance, watch these bombs being tested. And it's known that so-called downwinders, radiation subsequent to those atomic weapons, would come with the prevailing winds into southern and southwestern Utah. So one of the things we were interested, it turned out we were unable to test this question, was whether or not the families with the higher germline mutation rates were downwinders. And also, you know, not only does the slope, the rate of mutation increase vary as a function of age, but the y-intercept, the starting level of germline mutation, varies. And so one of the things we're really interested in is, to what degree does the age of onset and the duration of puberty have on sort of the basal level and subsequent levels of mutation. So I guess the high level thought that drives some of the work that we're doing now in the lab is, if the germline, the rate of accumulation of germline mutation is this variable in a rather homogeneous population from Utah. These are all European Americans, similar environmental exposures. You know, what does this say about the degree of variability that might exist across different ancestries and across highly differential exposures worldwide? So my initial thought was that environmental exposure probably plays an enormous role. And then this paper came out. Some of you may have seen it. Came out a couple months ago, but basically they looked at germline mutation rates in the offspring of Chernobyl worker survivors. Not just workers, but also people in the nearby area. And basically what they concluded is that there's really not any difference between the rate of the burden of germline mutation in the offspring of unaffected individuals versus those that were exposed to extreme radiation, which possibly suggests that those germ cells died off that were affected, or that really speaks to the degree of protection of the germline from exogenous mutagens. Okay, so what I want to close with is a follow-up story that was the result of a collaboration with Chip Stewart. I'm sorry, with Richard Coughlin, and he's really interested in what are the consequences of these higher germline mutation rates. And his hypothesis was that higher germline mutation rates predict higher somatic mutation rates in the same individual. And by extension, his prediction was that elevated germline mutation rates, because they're correlated with elevated somatic mutation rates in the same individual, would predict reduced lifespan in individuals with higher germline mutation rates. So we actually had a fairly unique opportunity to test this question with these pedigrees, because there were 33 different pedigrees, and in each of those pedigrees there's 4 grandparents. Those grandparents were born largely in the 1920s. Most of them have died at this point. And because of a database that we have in Utah called the Utah Population Database, we know when they died, and we know what's known about the cause of death. So what we did was we took each of those 122 grandparents, we looked at the germline mutations that they passed on to the second generation. Remember, we could attribute the mutations observed in the second generation to either grandpa's sperm or grandma's egg. We calculated a germline mutation rate for each of the 122 grandparents and stratified these grandparents by the highest quartile, middle quartiles, and lowest quartile of germline mutation rate. We had to do some corrections for their age of birth, the age of the parents, the age of birth for the parent of the offspring. But ultimately, when doing those corrections, we got these age-adjusted mutation rates. So let me break this down for you. The squares here are all the grandfathers. The circles are all the grandmothers. For each of the grandfathers, we stratified them into the 4 quartiles that I mentioned. The red squares are the grandfathers with the highest quartile of age-adjusted germline mutation rates, and the blue squares are the lowest. And similarly, for the grandmothers, we have the highest germline mutation rate individuals and the lowest germline mutation rate individuals. Then what we do once we have those 4 strata of germline mutation rate, age-adjusted germline mutation rates, we could go back to the Utah Population Database and correlate those germline mutation rates. We basically do a survival analysis. So we worked with a team at the Utah Population Database led by a great collaborator named Ken Smith, and this is the result. What we find, the individuals with the highest germline mutation rate in red, in the 75th percentile or higher, have a markedly different survival probability than those with the lowest germline mutation rate. So the hazard ratio is just over 2, and the median survival difference between men and women in the 75th percentile germline mutation rate versus 25th percentile is about 4.7 years. So this quartile has an expected lifespan of about 4.7 years versus the individuals with the highest germline mutation. So a couple of caveats. So first of all, the intuition. So the idea is that these individuals with the highest germline mutation rate also have higher somatic mutation rates, leading to a greater rate of organ senescence and reduced longevity. Important caveat is that there's only 122 individuals in this study. So 61 grandfathers, 61 grandmothers. And so each quartile only contains 15 individuals. So we have been trying to work closely with the DCODE group to replicate this study. We have not convinced them to try and replicate this, but by our understanding, they have all the data in hand to replicate our relatively small study. But the significance of these survival differences led us to write a grant last year, which really seeks to directly test the hypothesis that I just mentioned, namely that elevated germline mutation rates predict elevated somatic mutation rates. That's what we're inferring, and that follows the somatic theory of aging. So what we're doing now, we just got this grant funded using a program in Utah called Donor Connect. We're able to do rapid autopsies on individuals who've recently died. We can collect testis from those individuals as well as somatic tissue from different organs. And using some new sequencing strategies, we can directly compare germline mutation rate by looking at differentiated and progenitor cells in the testis versus somatic cells. And what we predict is that, yes, there'd be this correlation between an individual's germline mutation rate and somatic mutation rate, ideally connecting the dots between underlying the survival analysis that I just mentioned. I really emphasize that this study needs to be replicated. It's small, it's admittedly small, but even with a small sample size, it's statistically significant. And so with that, I just want to wrap up and thank again Tom Sassani, who now is did a postdoc with Kelly Harris at University of Washington and is now a staff scientist at Recursion in Utah, as well as Ken Smith, who helped us with the Utah population database queries to do those survival analyses. And I'd also like to emphasize, if you're interested in this type of work at all, the mutation data and all of the analyses are available at this GitHub link. There's some Jupyter notebooks showing all the analyses and the raw data underlying all of our analysis. So it's available if you want to try and replicate it or explore new questions. And I also like to thank all the funders who supported this work. And I hope I've left time for some questions.
That was great. Thank you very much, Aaron. Really awesome talk. You have a question from Boxen Zhao at Boston Children's. So the question is about the number of de novo mutations in paternal age at birth. So is it possible to further classify the de novo mutations in the third generation into paternal and maternal origins? And I think a plot showing DNA mutation from father and paternal age at birth will be more accurate.
Yeah, so to the first question about whether or not we can stratify mutations in third generation by gamete of origin, yes, but not nearly as comprehensively as we can the second generation. So essentially in the second generation, I skipped over these methods, but in the second generation, we can use linkage, a haplotype linkage from across the 3 generations to attribute mutations to the grandparent of origin about 95, 96% of the time. In the third generation, we're unable to do that. The way we have to attribute mutations to mother or father is by something called read mapping. So if we have a read-based mapping, so if we have a sequencing read that overlaps the mutation, we're doing paired end sequencing. So often we have 500 base pair fragments. Some of the time, the other end of the fragment will overlap an inherited variant from one parent. So a situation where one parent is homozygous and the other parent is heterozygous. If that other end of the fragment overlaps that heterozygous allele, we can attribute the de novo mutation by linkage to that parent. That only works 25% of the time. So to answer a long-winded way of saying, yes, we can do that in the third generation, but only for about 25 to 26% of the mutations. And second point, I think a plot showing de novo mutation from father instead of, sorry, I don't quite follow that difference. Maybe you could follow up with me offline about that one, or if you want to speak up, I don't quite understand the distinction in the plot.
Right, we can unmute you if you. I think my understanding is that so that plot is basically for the paternal age. So is that okay to plot separately, like for mother's age and the dad's age, versus the DNMs, total GMS? I think that's what I understanding, but I can unmute you to clarify that.
So the plot was, and I apologize if the labels weren't clear enough. For each point is a child, and the Y value is the number of mutations observed in that child, and the X value is the parental age at the birth of that individual child.
Thank you. Do we have other questions? I have a question. I was looking at your 2019 paper talking about the constrained coding regions.
Yeah.
Can you address the mutation rate with respect to the constrained coding regions?
Yeah, that's a good question. So the constrained coding regions, the whole idea there was inferring regions where there is mutation, but we don't see genetic variation, or we don't see mutations in those regions when we study healthy individuals. And the idea is that those are regions that are enriched for de novo mutation in developmental disorder individuals, or one of the hypotheses. There are some regions, especially of ion transport domain proteins and some other relevant proteins, where we don't see a single mutation for more than 100 base pairs in 300,000 people that are studied. And the caveat being that those 300,000 people are known to not have developmental disorders, so they're sort of a control. So what we don't actually know what the mutation rate is in those regions. I actually expect the mutation rate in gametes in those regions to be the same. It's just there's an ascertainment bias. We never get to see mutations in those regions because of how deleterious mutations therein are, if that helps.
Yep.
So it's actually, I don't know if any of you are attending AJHG this week, but there's a poster from Caitlin Samoha, who's a new faculty member at MGH, I think. She did some competing work in that area at the Broad Institute as part of her PhD. As part of her lab now, she's got sort of a follow-up study to our work that her student is presenting at AJHG from sort of, I think, 400,000 individuals, whereas our initial study was on the order of 120,000 individuals.
And there's one more question, Aaron, in Q&A, if you can see that.
Oh yeah, okay, so right, yeah, absolutely. I can do that, bear with me. Just gotta remember where that slide is. Right. So the thought experiment here is, sorry, I'm just trying to clear out my screen here. If this is truly a de novo mutation, the second generation, and not a sequencing error or something technical that we don't care about, then this T mutation is present in 50% of this dad's sperm. The A variant, A allele is present in the other 50%. So this is very much like flipping a coin, heads and tails. That we're diploid. So at every site that we are heterozygous in the genome, our sperm, if you're a man, or egg, if you're a woman, should have 50% of those gametes should have one allele, 50% of the other should have the other allele. So it's like flipping a coin, probability of heads versus probability of tails. Therefore, it can be modeled with what's called a binomial distribution or a binomial expectation. So the idea is, if this is truly a real mutation that's present in 50% of sperm, Therefore, that mutation should be, the expectation is it's present in 50% of children, of offspring of that individual. But that's the expected value. So just like we expect 50% of coin flips to be heads, go flip 8 coins, do that a bunch of times, and you'll find that, you know, a lot of times you get 1 head or 2 or 0. So this expectation is, if it's true, if it's truly a de novo mutation, if there's 8 offspring, the binomial tells you that if the probability of success or seeing this T allele is 0.5 or 50%, the probability of not seeing any of the children is 0.5 to the 8th, just as not seeing any heads in 8 coin flips is 0.5 to the 8th. Conversely, therefore, the probability of seeing that mutation or heads in 8 coin flips, and at least one of 8 coin flips, is 1 minus that probability. I hope that clears up. If not, I'm happy to elaborate.
That's great. Okay, and we're on time too. Well, thank you very much for coming and presenting. Great talk.
Absolutely, yeah, no, I really appreciate it. It was a pleasure, and nice to meet you all.
Thank you so much. Really nice talk. I'll see you guys next time.
Great, take care.
Have a good day.
Bye.